So welcome to the fall 2021 Viromics Workshop, which was going to be in person. We're going to host it here as a webinar series, both because we wanted to reach a broader audience, but also just in light of varying levels of risk across the countries that we hope will be joining us today. I'm Matt Sullivan, and I'm helping host this. I want to just reach out right away to thank Olivia Zablocki and Britt Fauner, who have both been working behind the scenes to make sure that we're ready technically, and they both are. This is their first big big Zoom platform experience. So we're learning together, and you can see a number of the panelists will be involved with speaking over the next couple of days. I'm going to open today as I share my screen. I'm going to open today with a half an hour describing sort of an introduction in the background to get us started. So I wanted to first just emphasize that these Viromics workshops, this will be recorded today. You're part of the live audience. I hope you're ready to be engaged. We've left an hour out of this time to be really discussion opportunity with the particular speakers of that day. So we'll be looking forward to your questions, and the moderator will handle those questions and work with the panelists on getting those answered. But I also wanted to emphasize that we've been doing a number of Viromics workshops through the years. We started with what I'll call environmental virology. It was more broad than Viromics back at the Biosphere when I was at University of Arizona a number of years ago. And we have since done 4 Viromics workshops at Ohio State. This is one of the most recent ones. We typically try to have between 30 and 40 early career folks who come to these, and then a handful of teachers. These are great opportunities for in-person networking. And while we intended this one to be in person, we're not quite sure it's ready to be. So next May is our tentative date for the next in-person Viromics workshop at Ohio State again. And as a workaround in the near time, we did do a microbiome informatics webinar series that represents 2 to 4 hour hands-on working group sessions that are led by all the great early career researchers here that went through microbiome science in general. And I'll just focus on one component that you'll most be interested in, the Viromics component that Olivia Zablocki and Ben Bolduc covered towards the tail end of that microbiome informatics webinar series. Those are all archived and recorded and available on the Ohio State website as well. But today we're here as part of the first of a 3-day Viromics workshop. And as I mentioned, it's now obviously virtual. How do we get here? This is the steering committee who helped me organize this. This is a great group of folks who offered a lot of feedback and really iterated with me on actually an in-person agenda. And then when I made the call to shift to virtual after consultation with them, really revising it into this format that you're seeing today. And I would like to give a shout out to our sponsors. The NSF funded part of this, as well as the Ohio State Center of Microbiome Science. So some of the people power to get this ready supports come from sponsors. So thanks to the organizing committee for great discussions so far. Some of them we're going to hear about from in their expert talks here today, tomorrow, and Friday. And I look forward to working with them and others in the future organizations of this Viromics workshop. So what are we going to try to do here in the next 25 or so minutes? First, I want to get us excited about why we should study viruses, and particularly microbes, because that's where the science is at now. And then just share some stories about what Viromics is teaching us right now, with a focus mostly on the oceans and just hints at the soils and humans. It is focused on my lab's work, but we'll mention some of the other highlights from other groups that towards the end as well. And I'll be mentioning at the end sort of the structure of this webinar series, what you can expect from the next few days. So why should we study viruses? Microbes, we hear about the human microbiome all the time. So I start here, the idea that microbes in our body are as abundant as human cells. These microbes do all kinds of things to interact with our body in the human ecosystem to impact our health and disease status. But beyond that, microbes are critical in soils, feeding plants or as pathogens of plants, so they can impact agriculture yields. And if you care about breathing, and I'm sure most of you do, half of the oxygen that you breathe in comes from microbes. It's marine photosynthesized throughout the vast regions of the oceans that cover our planet. And what I'm interested in focusing on is sort of what viruses do to sort of manipulate those microbial impacts, those microbiomes and ecosystems throughout human soils and the oceans. And if we focus just on the oceans, we actually didn't think there were many viruses for many years, and groundbreaking work from Curtis Suttle and Jed Furman, Eric Womack, and Karina Broussard, and many others started to realize that actually, if we concentrate seawater and look at it, this is a cyber green DNA stain. You'll see big dots, which are cells, and little dots, which are viruses. There's a lot of viruses there. If you swallow a mouthful of seawater, you swallowed 50 million viruses. If you do tracer experiments, you can see in bottle experiments that about 1/3 of cells are killed per day due to viruses. We can't culture many of these, far less than 1%. That's because we can't culture their hosts. And these play important roles in moving genes around. Viruses transduce genes from one cell to another. And there's been very few measurements, but if we scale up from a Florida estuary number to the globe, there'd be something like 10 to the 29th genes moved per day. A lot of times people talk about Bordetella toxin, Shiga toxin, pertussis toxin, cholera toxin as microbial toxins, but it's actually the virus integrated in the microbial genome that is encoding the toxin gene. And so a big challenge for the field, once we realize there's so many, is how do we actually assess their ecosystem impacts? And I didn't like counting dots. But the problem was that we didn't have Viromics yet. And we didn't have it, I would suggest, because of this kind of problem. So if you're to use sequencing and you go out and survey viruses from the environment, everything, or we might use in ecology or evolution the term population. And this was a problem because the paradigm was that virus genomes are rampantly evolving. And if they're rampantly mosaic, then we might expect a continuum in sequence space. So this is virus sequence space, just pictorialized. And on the left is the idea that there's a continuum in that virus sequence space. So in this scenario, a yellow virus might exchange a lot of genes with a green virus to where there's yellow-green viruses that are hard to distinguish from the yellow or the green viruses, which blend into the dark green viruses, which blend into the blue viruses. And so it would be very hard to sort of identify a unit. And if we're doing ecology or evolution, what we'd really like would be some structure in the sequence space, discrete populations, where the yellow virus is different than the blue and the green. And I would say that though that paradigm existed, it was really from viruses that have been studied evolutionarily, which to a large degree were single-stranded DNA and RNA viruses, which happen to have very high mutation rates and recombination rates, and much higher than double-stranded DNA viruses. And so we went out and tried to actually ask the question, well, what does viral sequence space look like in a natural system? And so this is in seawater for viruses that infect a cyanobacterium called Synechococcus. And actually, this sequence space representation is cyanophage genomes that I spent 10 years on many different hosts and many different seawater samples, isolating, characterizing, and sequencing. So this is my poor productivity of 10 years of PhD and postdoc work. And when we did a 10-minute experiment with a new method, viral tagging, we actually could look at a lot of these at once. And the red dots are viruses that were captured in viral tagging. And we saw data from 10 to the 7th viruses in a single 10-minute experiment. And what's nice is we also got variation at the same time. So if you're looking at 10 to the 7th viruses at once, you can look at the variation. And so the clouds that I've just put up, those clouds are the resampling of the population genomic variation in the data set. And what you can see is that their structure, they don't overlap. And in any place that you think they do overlap, actually, it turns out when you look at the end of the page axis, they do not overlap. So this suggested that viral sequence space, at least for cyanophage, is clustered. And so we could use empirically determined a 95% average nucleotide identity to be able to identify populations in natural systems. And follow-on work from Anne Gregory, who isolated 142 cyanophage genomes from half from each of 2 sites, sequenced all the Genome Institute through their community sequencing program. She showed using formal population genetic metrics that gene flow is higher within and between these groups, and that there's selection that's different across the different populations. So formally, these populations, at least in the case of cyanophage, look like species. And then Melissa Duhame looked at pseudo-Alteromonas phages and showed that in pseudo-Alteromonas, when she phenotyped them, looking at host range as a metric of phenotype, she found that these populations were consistent with the kinds of host ranges that she saw. So there are two different ways to look at sort of ecological readouts, if you will, of the underlying structure. So we now have a way to count, or a unit to measure of these viruses. So what is Viromics teaching us? And I said I'll focus in the oceans, and the oceans will be three kind of vignettes: patterns, processes, and paradigms. And to do this work, I was very fortunate to join the Tar Oceans Consortium, which were doing global sampling on a sailboat using standardized approaches, and not just viruses, but viruses to fish larvae. In fact, across 8 orders of magnitude in ocean biota size on a sailboat. And this really is our research vessel, converted to be able to put all kinds of really interesting instrumentation on it. What do we learn? Not surprising to any of you, if you're here for a Viromics workshop, everything was really new. So 90% of what we were seeing was brand new. And this meant we really just started with catalogs. So when we started, there were about 2,000 known virus genomes, viruses that infect microbes. And after some of the early work, we grew that 15-fold to 30,000 viruses. And more recently, in a single paper, we added 200,000 new, and these are not virus genomes, actually higher number of genomes, but the number of virus species, which is that 95% ANI population definition. So we grew from 2,000 to 200,000, so a 100-fold increase. And you'll hear about vContact2. It's a gene sharing or machine learning approach for looking at genus level virus taxonomy. And so if we have catalogs, 200,000 of these entities now identified, we could use these to start to do genome-based tracking. And here, each gray dot is where the boat, the Tar Oceans boat, stopped for 24 to 48 hours. These are stations, and this is where sampling would happen. And the arrows here are, the colored arrows are virus flow. So if we follow population abundances from one site to another, which direction would we say viruses are going? And in almost all cases, it looks like viruses are following the flow of major ocean currents. And this is not surprising. We know viruses can't swim, so this is really not that interesting a result. But this genomic tracking was very interesting, and I'm going to come back to it in a little bit. But what it also did, as we mapped this ocean sequence space, is just helped us identify, well, which viruses are out there. And in particular, I was excited to see phage T4-like viruses. This is one, as most of you would know, that's a foundation of molecular biology. It's one of the earliest colophages ever studied. And that relative is all over the global oceans. But another one that was surprising was the third most abundant virus, and that's Phi38.1. And it just so happened we've been able to get that one into culture. And though it's not the foundation of molecular biology, it might be one of the most well-studied viruses to date because of great work that Christina Howard-Verona has been doing, where she uses time-resolved multi-omics data sets to look very carefully at the lifestyle, how this virus affects multiple hosts. Beyond sort of catalog maps, what kinds of processes has Viromics taught us about that viruses are manipulating? We already knew before Viromics came on the scene from cultures, cyanobacterial and their virus cultures, that viruses steal photosynthesis genes. And this was subject of a lot of work in the early 2000s, as you can see here. And the idea was that if we look here in the photosynthetic membrane, a photon of light could strike the photosystem, PS2 reaction center, that would split water to give oxygen and hydrogen, such an important process that 4 Nobel Prizes have been won studying that specifically. That excites and electrons pass through a series of electron donors and acceptors to the other photosystem, which is struck by a photon of light again, excites an electron. and goes off to ferredox and redox reactions. Now, the viruses steal that core reaction center protein, the cyanobacterial viruses. And the idea is that these cyanobacterial viruses require photosynthesis to continue, but that protein, because it makes oxygen from water, has oxygen radicals hitting it all the time. And so it turns over in most any photosynthetic cell on the scale of 30 minutes. The virus prevents the host from making a new version, usually because viruses globally shut down transcription and translation. So the hypothesis is the virus boosts the photosystem weak point to keep photosynthesis going by including that gene. But there's other genes. So PSBD is the heterodimeric mate to the PSBA. And so if you have a broad host range, it seems these viruses carry PSBD. And plasticinin and ferredoxin are rate limiting steps of electron flow away from photosystem 1, so keep it from damaging. And the highlight inducible genes are thought to protect PS2. So we think that viruses are probably randomly stealing genes, and the ones that cause a fitness advantage during virus infection stick. And so we asked them with Viromics what other processes are viruses manipulating, and this is where Bonnie Hurwitz discovered 35 central carbon metabolism genes. A number of groups, including two of our speakers, Simon Rue and Karthik Anantharan, have discovered genes involved in sulfur cycling, and many genes involved in nitrogen cycling. So viruses are manipulating microbial metabolisms directly by encoding these auxiliary metabolic genes. And this is a very broad phenomenon in the environment. So those are processes that viruses are impacting. What paradigms are we starting to revise? So we'll come back now to the result I suggested wasn't that interesting. This is if we look at the data laterally. If we look at the data vertically, where these X's come in, the X's are where the viruses appear to be sinking. Why would they sink? Viruses shouldn't sink. And so the hypothesis is this. We think the paradigm has been that bacteria and phytoplankton are infected by viruses. This produces organic matter that gets recycled through this process called the viral shunt. And so viruses should keep carbon small in the ocean and not contribute much to the broad biological carbon pump that the oceans are so important for. But instead, what if viruses actually, as a local explosive product, create aggregates, and those aggregates could then sink out of the water column with viruses embedded as export material? Or what if virus-infected cells are preferentially grazed? That might also lead to, there's data for and against this particular hypothesis. And so in this case, you could imagine viruses impacting carbon flux out of the surface ocean. So we decided to test this with what we call a genes to ecosystems modeling approach, where you're using sequence-based data, large-scale compute, and ecosystem rules, combining machine learning of large-scale data sets with ecosystem modeling, and ask this question: which organisms most impact carbon flux in the oceans? And so Tara, what was unique is they had a first measurement through the global oceans along that cruise track and through depth of carbon flux. And the blue is areas of low carbon flux. And the red are areas of high carbon sinking from the surface to the deep ocean. And so though we looked at data from all the different size fractions, viruses, prokaryotes, eukaryotes, viruses better than any other organisms predicted this carbon flux. In fact, they predicted 89% of the variation of carbon flux. So this supports the idea that viral lysis does help form aggregates, and this may lead to export. And maybe this means that viruses could be used to tweak the ocean carbon sinking capacity of the global oceans. And I just mentioned this because this approach, which was developed by the Tarjan's consortium in the Guidi et al. Paper, is really generalizable. We asked the question here for ocean carbon flux, but you could ask the same question in disease, soil health, or in product yields. I also want to just flag, all the analytics that are developed are available now on a collaborative cyber infrastructure that's funded by NSF, and another one, many of them on a DOE-funded cyber infrastructure, Cyverse and KBase. And as part of the iVirus ecosystem of apps that Ben Voldick, Simon Rubani Hurwitz, and others have helped develop through the years. So iVirus is this community available toolkit to be able to do these kinds of viromic analyses. So that was the oceans. And just to briefly touch base on the idea that this Viromics toolkit is helpful in other ecosystems, what about soils? And so we'll focus on one of my favorites here, a model ecosystem called Stordalen Mire of climate change up in northern Sweden. There's long-term research station there, lots of instrumentation to measure ecosystem fluxes, and it's an area where there's clearly active permafrost thaw happening. This is one of the walkways to get out onto the permafrost that's sunk in as a result of thaw. This is part of a new eMERGE Biology Integration Institute that's funded by the NSF. And we had a lot of bulk metagenomes that were sequenced through work from Gene Tyson's lab. And one of our speakers, Friday, Joanne Emerson, will be moderating that session. And she led the work to look at viruses in those bulk metagenomes. And so what she found, and what Gary Trubel, a student at the time, found in related analyses, was that there's a lot of soil viruses. They're present. We doubled the number of known soil genera. The populations appear to be active, at least based on metatranscriptomic inferences. These viruses are predicted to infect key carbon cycling microbes, and they have carbon cycling AMGs. So Joanne's continued that work in her own lab and other soil systems, but this is a great example of soil viromics in action. In humans, we just remind ourselves that we're as much microbial as we are human. There's as many microbial cells as human cells, and in fact, there's 100 times more microbial genes than human genes. And of course, I'm interested in what are the role of the viruses in these systems. And so this led us, when we started to do our work, the first issue we were having is that there weren't good databases available. So Anne Gregory with Olivier Zablocki screened all available microbial metagenomes and viral metagenomes at the time, it's 2,700, and pulled out 33,000 viral populations. There's a lot more genomes there, but when we dereplicate those into our 95% ANI populations, is 33,000. And they used that to show first that you had a better chance of predicting the kind of viral community that's there based on whose lab prepared the sample, because of the methods they used, than sometimes the disease state. So the way you process a sample impacts the resultant virome, really important. In oceans, we've sort of all transitioned to a systematic approach. And then they also looked at how viral richness changes through time in aging populations. So this was nice work for us to set the stage for our other human virus work. And I'll just flag that and Olivier did this work, but there's been at least two other groups that have really pushed hard and pulled out a lot more viruses. So in the human space, there's been a lot by two other groups, and one of those is Rob Finn, who will speak on Friday. Another way viruses could impact the humans is through this idea that a complex disease may actually have an ecology associated with it due to the microbial and viral underpinnings. And as an example of that, we were interested in looking with collaborators Greg Caparasso, Jim Adams, and Rosa Kamalnik-Brown. We're interested in looking at autism, where in the gut, it was known that bacterial diversity decreases and fermenting bacterial diversity decreases, but viruses were not known. And so we were interested in experimental design where we looked at adding microbiota through fecal microbiota transplant, in fact, through a sustained FMT treatment. And what we saw is that autistic children's GI symptoms went down drastically. 16 to 18 children improved. Please note the numbers. This is a pilot scale open label study. So it needs to be done more broadly. But 16 of 18 children improved as measured by behavioral autistic symptoms. And we were able to map the viruses and bacteria that correlate with these improvements. And as I mentioned earlier, there's that ecosystems modeling approach, machine learning and ecosystem modeling approach. Ecology allows us to see the thousands of microbes and viruses in the gut, and those approaches can actually identify the few that matter. So we didn't do it at the time because that was a number of years ago, but we could go back now and take that ecosystem approach to identify the few that should be followed up and studied. And that's in autism. We've also got the work I mentioned on aging. We're looking at phage therapy, smoking, and spinal cord injury as other systems where we've continued to do work and apply the Viromic approaches. I focus on my lab's work because I know that the best. But just reviewing the literature the last couple of weeks, and certainly just as I've read through the years, it's just been amazing to see some incredible stories coming out. So I just want to give a shout out to some of the, for me, personal highlights of stuff I've seen from other groups out there. So the first is just, viruses going big. And so there was some beautiful work from Friedrich Schultz and collaborators at Joint Genome Institute, where they looked at giant viruses and just really blew apart what we're seeing, as well as huge phages work that was published where they're looking at larger phages. In this particular case, they had also the interesting story of multiple CRISPR-Cas systems that were identified and some nice stories revolving around that. Related to discovery, I think Christina Moraro, who will be speaking, I believe, tomorrow, shows a great example of how you can find a new virus and instantly these days biogeographically contextualize it, at least if you're in the oceans or other systems where there's broad data like this. She discovered the covoviruses and put those on the map, quite literally, right away. And so her work in this ISME journal paper shows that biogeographic context for the new virus group, the covoviruses. And then some nice work from Evelyn Andreessen, who will also be speaking. She's going tomorrow, but she's moderating that session. Shows some nice work looking at ecosystem connectivity and the idea that you can kind of use that viruses as genomic tracers, but in a different context here to look at the penetrance of viromes that may come out of wastewater and where do they end up in other aquatic systems that are linked to that wastewater treatment. In the human space, there's beautiful work from Colin Hill's group that's now showing that the human gut virome is highly diverse, stable, and individual, and that's sort of a very clear, clean message from that data. And I thought there's some nice work reviewed by Rick Bushman in a Nature Reviews micro paper where they look at a lot of things, but one particular figure jumped out for me is very interesting because the human microbiome and now human virome space tends to be really good at incorporating time into their analyses. And so there's some nice work showing longitudinal changes in infants and how the virome changes through time. Lots of other great virome science out there. I don't mean to skim over anything else. I just wanted to touch base on some personal highlights there. So we're all here though for this webinar series, not to hear me drone on and on. We've got 3 days of great action that's focused from 1 to 3:30 p.m. Eastern Standard Time, wherever you are in the world, the different time frame. It'll be the same time each day. It's going to start with a half an hour introduction and then about a 15 to 20 minutes left for each speaker. We'll see if they can keep to a few minutes extra for questions. We're going to hear from 3 speakers today and each day. Today's are focused on virus identification tools. The second day is focused on virus classification tools. And the third day is focused on databases. Oh, and I forgot to mention, after the speakers at 2:30, the last hour is a roundtable. We'll want your questions. I'll be, as moderator, looking for your questions to identify the things to feed to the panelists. So please put into the chat your questions for the roundtable so we can have a nice discussion. Again, the second day and third day, classification and databases. This will be moderated by Evelyn and Joanne, respectively. So in summary, I realize that was pretty quick. We've talked about quantitative systematic sampling and how important that can be to creating large-scale opportunities for science. The idea that we can even create a catalog. We now understand the unit of a population or species and genus level units through different tools. We can measure those genus level units through the machine learning powered gene sharing networks. And once you have those units that you can count, you can do ecology. And in this case, we used genes to ecosystems modeling on a global ocean carbon flux data set to show that viruses are driving carbon flux in the oceans. I also mentioned that Joanne's work and Gary Trubel's work have been really impactful in the soils at this thawing permafrost site. There's a lot of other great soils Viromics starting to come out as well. and the idea that Viromics is really penetrating a lot in the human space. I want to put a plug out to my home institution. Ohio State has two new big well-funded entities, the Center of Microbiome Science, which is creating a lot of tools and resources, as well as the funded Emerging Integration Institute, both of which are going to help really push the sort of multi-omics data integration space quite a bit. Thank our sponsors again. And I hope it's okay, I mentioned we have an open postdoc and graduate student position. So please contact me if you're excited about this kind of work and would like to apply to do something with us.